Cloning has been a technique scientists use to make exact genetic replicas of something. They noticed that cells, genes, tissues, and whole animals can be cloned. What is funny that cloning actually already exists, such as bacteria. Bacteria makes exact copies of themselves and spread. Different animals that were cloned before were sheep and plants. The first ever successful cloning was done in 1952, which was a tadpole. Then in 1963, the first clone fish was created. Then in 1970, the first genetically identical mouse was produced. Then around the 1980s, the first sheep and cattle were cloned. Well, you may ask yourself, how is cloning done? Well, the first method we are going to get into is embryo cloning. An embryo is an organism in its early developmental life cycle. They are fertilized eggs that have begun cell division and can be up to 8 weeks old. Embryo cloning is when an embryo is split into two and they placed into a mother's uterus with those two animals developing and sharing the same genes. Scientists start by removing a bit of the DNA. Then the DNA goes into a cell wall and is grown into an embryo and transferred to the circuit of a mother. Another method of cloning is called nuclear transferring. The somatic cells are what makes up an organism, excluding the sperms and egg cells. What scientists do is that somatic cells already contain two sets of chromosomes, and they transfer the DNA into an egg cell, then let it develop into an embryo, then finally putting it into a mother's uterus. The first mammal to be successfully cloned by nuclear transferring was a sheep named Dolly. In 1966, she was cloned by a Scottish scientist. She was cloned by taking an udder cell from an adult sheep, and it took around 276 times to get Dolly right. The goal for making Dolly was to make a better method in developing modified livestock. Dolly was taken from the gland of a Finn Dorset sheep, and the egg was taken from a Scottish blackface. When she was born, they had the identical face of her mother. Dolly now is in the National Museum of Scotland on display. The first testing done to create a clone was in the early 1900s by a German scientist named Hans Spemann. He tested it on a salamander embryo and was awarded the Nobel Prize for Physiology or Medicine in 1935. He later theorized that there was another way to clone organisms, not just by embryos. He called it nuclear transferring, and when it was done in 1952 by two American scientists, they used DNA from embryonic cells of the frog type called Rhinopipians to create a cloned tadpole. Finally, in 1958, British biologist John Bertram Gurdon successfully carried out a nuclear cloning by transferring the DNA of an adult intestinal cells of African clawed frogs. He was awarded a share of 2012 Nobel Prize for Physiology or Medicine during a breakthrough in his works. The difficulties in cloning are that organisms have health effects, and it includes birdsides, defects in vital organs such as the liver, brain, and heart. There also have been effects of premature aging and also problems with their immune system. There can be many genetic changes that occur during the embryo development which can cause these problems. Scientists were able to clone human embryos for 7 years now, but not a whole person. This is because of ethics as well as how difficult it would be to actually clone human. They will need to fix genetic issues and keep retrying with humans to the point where it's unethical anymore. There is a lot of problems in cloning though, as there can be many complications to genetic mistakes. Nowadays, scientists are using cloning to make a stem cell factory, which is a cell that can create tissue and help it grow back. Thank you.